Okay, then the second talk is also by Valery Korneyev, but it's a work done by a big group of people. It's a discovery and revision of the bamboo picture wing flies from family Ulidiidae.
Sorry, I will be reading again.
That's not Tephritoidea. It's Tephritoids.
They are tephritoids, still tephritoids, even after all these years. So I will try to find my laser pointer again. Okay. So this talk is about a recently discovered diversity of picture wing flies associated with bamboos in South America, recently discovered by Damir Kovac and John Smith from the Naturalis Leiden. The main contributor for this talk was Yelena Kameneva, but she asked me to present her work, and we decided that I would be a talking head, although I was actually only the illustrator and coordinator of this paper. More than 110 years ago, Friedrich Händel described several unusual picture-winged flies from Bolivia and Brazil, mostly based on material from the Schnuse and Garleb expeditions. They had bizarre wing venation with an extremely shortened pterostigma and a lobed costal cell, similar to what we know in the Afrotropical schistopterine taphrites, as shown here. Another unusual picture wing fly related to our talk was Ulidiidae inversa, described by Speiser from Brazil. Its syntypes deposited in Königsberg were destroyed during the Second World War after the occupation of Eastern Prussia by the Soviets. Ulidiidae differs from the other Lipsanines by the very basal position of the radiomedial wing, as you can see on the lower picture here, very basally. Most of them remained known only from the type specimens and the original description until Damir Kovac and John Smith collected some very peculiar flies on bamboo in Bolivia and Peru in 2009 and 2011. John Smith observed flies on bamboo combs, and they were certainly Aspistomella lobioptera. And another species was very similar to Ulidiidae inversa. Damir Kovac found several other species of Epocytin into holes made in bamboo combs by the bipearlite caterpillars. He observed the fly larvae feeding on the bacterial film inside the comb. These larvae were able to swim above water with their posterior spiracles exposed. He was able to rear some adult flies from the larvae and described the morphology of the larvae. Kovac sent this material to us, to Yelena and me, for identification, and this epic taxonomic revision was begun. They represented 4 species apparently belonging to Aspistomella, and Ulidiidae. All 4 species were previously undescribed. Damir Kovac made excellent scanning electronic photographs for some adults along with detailed descriptions of the larvae. Only one of them was actually a named species, Aspistomella heteroptera. The other 4 species were new and unnamed. We give here some manuscript names, for instance, Aspistomella duo, Aspistomella tres, Ulidiidae arcuata, Ulidiidae tenoris. Very similar, sinuate structure of the larval posterior spiracles and extremely narrow mouth hooks are actually believed to be synapomorphisms of Aspistomella and Ulidiidae, as they are pretty uncommon or doesn't occur anywhere in the Ulidiidae family. So adults of which of these 2 genera otherwise represent extreme morphological forms within this group of genera. To discovery of species similar to Aspistomella lobioptera, but different in the structure of the sestile and phylae. The depth of the costal cleft and the length of the pterostigma vary from Lobioptera to the species formerly described as Euxesta lunata handle, where they are almost unchanged, just in a basal state. In addition, Aspistomella pachytea shows another morphologically extreme form, which has a linear, rudimentary pterostigma, but neither the cleft on the costa nor a lobe of the costal cell. 4 new species from Bolivia and Brazil are added to the genus Ulidiidae, but 3 of the 5 are known from a single specimen and a single sex. Ulidiidae arcuata shows sexual dimorphism in wing coloration and venation. Species assigned to Paraphiola, Polyteloptera, and 6 additional new species are generally similar but show considerable morphological diversity and can be placed either in all genera Aspistomella or Paraphiola, Polyteloptera, and so on, or in another new genus, Catulivellia. But an attempt of phylogenetic analysis did not give a satisfactory solution. Most new species, except a few, are known only from females. Male genitalia also do not provide essential diagnostic characters. Species of Aspistomella group of genera, however, can be distinguished from other Lipsanini, by their physiognomics. They have heads with elongated, anteriorly produced epistome combined with a very low clypeus. The most important character which gave an idea of the position of the Aspistomella group within the tribe Lipsanini was discovered unintentionally while taking a high-resolution picture on the fronts of Aspistomella lobioptera, namely a strong differentiation of the row of strong frontal city here. They are strong almost as in Tephritoids, and tiny parafrontal setuli lateral of them here. This is a spoiler, in fact, for the results of another project started by Elena Kameneva in Berlin, a morphology-based phylogenetic analysis of the tribe Lipsanini revealed an important reconsideration of generic groups. Two main lineages, Metisa lineage and Euxesta lineage, contain the vast majority of the tribe's species. I'm sorry, I'll be back to show you this picture. This is important because on the first picture you can see the status of the character which occurs in the most Euxesta and related species, but half of Euxesta and other species are like here, and they represent a whole lineage to which Aspistomella belongs. This agrees pretty well with some unpublished results of a multi-gene molecular analysis presented by Charles Wallis in his master thesis 6 years ago. The Amethysa complex includes all species mainly associated with tall grasses, Poaceae, including corn and bamboo. The species of the Euxesta complex are more generalized consumers of decaying plant material, including some palms and even dicots infected by fungi, etc. The Aspistomella lineage appears as such a monophyletic cluster within the Amethysa complex. So the conclusions. The Aspistomella group contains about 23 species, 19 of which are new. They may form a monophyletic lineage within the Amethysa complex of genera. Most, if not all, species of the Amethysa complex have larvae that live in grasses, often in tall grasses, and include all species that infest sweet corn or maize. The Aspistomella group of species includes all known Lipsanine species associated with bamboo. But this generalization to other species is by far premature. We hope that revision will stimulate further study of bamboo flies in South America. We appreciate the support of the Philip Schwartz Institute for funding Elena Kameneva project and the curators who provided important collection material. And thank you all for your attention and standing for our country.
Thank you, Valery, for this really interesting talk. And please, if somebody has questions, we still have time for at least one. Yeah, Keith.
Some of the most morphologically divergent, bizarre tephridids in the whole world in Australasia are associated with bamboo. What do you think is unique about that habitat?
They look to be similar. The problem that bamboos are relatively new compared to the early, say, Eocene forests, which then turned into tropical rainforests. So bamboos usually replace the cut forests, so they appear. So these groups completely very distantly related and completely have nothing to do with each other, but they take almost the same niche in the same way. But oriental bamboos are usually cut by weevils, as was shown also by Damir Kovac and Amnon Freiberg. And the non-tropicals are mostly eaten by the caterpillars. That's the only difference. So the difference is in the insects which make the holes for them.
Oh, please, Alan.
Other on their own bamboo.
Sorry, again.
Are there other Ulidiids that infest bamboo?
Ah, sure, there are Reno, but this is absolutely different. And there is no documented feeding. They are occurring in association with bamboos. They are very numerous, but nothing is known exactly about their Reno biology. Maybe not all of them, but most of them are associated somehow with bamboos. This is a different tribe and probably a different variety. Why not? Why not? Bamboos are good niche. Why not twice? Why not invest twice, even in the non-tropics?
Oh, thank you very much. And